Thank you, Dr. Weiss, and hello, everyone. My name is Ryan Severja. I'm one of the chief residents for the internal medicine program, and it's going to be my pleasure today to be introducing our speaker, Dr. Lanata. So Dr. Lanata earned her medical degree from Universidad Peruana Cayetano in Lima, Peru, and completed her internal medicine residency program at Georgetown University Washington Hospital Center, where she also served as chief resident from 2012 to 2013. She went on to complete a fellowship in rheumatology at the University of California in San Francisco in 2016. And then from 2016 to 2021, she was an assistant professor of medicine in the Division of Rheumatology at UCSF. In 2021, she joined the Genomics of Autoimmune Rheumatic Disease section at the National Human Genome Research Institute, and she is a nationally and internationally recognized expert in population genomics and autoimmunity. She has particular expertise in systemic lupus erythematosus, and she has extensive experience leading and collaborating on large patient cohorts and has served as a co-investigator for the California Lupus Epidemiology Study. That study has generated multiple transdisciplinary research initiatives as well. She currently serves on the SLE Gene Curation Expert Panel for ClinGen, the clinical genome resource, and is contributing to efforts that define the clinical relevance of genes and variants for precision medicine. Her work has been recognized with several prestigious awards, including the Scientist Development Award in 2017, the Gilead Scientist Scholar Rheumatology Award in 2020, and the NIH Diversity Supplement to an R01 in 2020 as well. And finally, the Rheumatology Research Foundation Investigator Award in 2021. So please join me in welcoming Dr. Lanata.
Thank you so much for having me. I'm really sorry not to be there, and this is a wonderful invitation. And I'm here to talk about the promise of personalized medicine in rheumatology. So the outlines of today is really understand the challenges of what it is to be a rheumatologist these days, and what is disease heterogeneity, the promise of multiomics and the current research agenda, and what we're looking forward in the future in clinical care. So I wanted to start with a case that happens very frequently. I'm sure most have encountered this type of patient, like a young 26-year-old female who's admitted to the hospital with skin rashes, anemia, thrombocytopenia, inflammatory arthritis, and frank proteinuria. She looks a little ill, a little high blood pressure, and she has a clear raised erythematous macular facial rash, as you can see in the picture, that spares the nasolabial fold, very compatible with malar rash. An exam, we can also see evidence of synovitis and laxity of joints. And her labs are compatible with anemia, thrombocytopenia, low leukocyte lymphocyte count, elevated creatinine in 1.1, low albumin. You look at the UA and it shows hematuria and pyuria, and in a spot UPC reveals 1.8 grams of protein with high inflammatory markers. And here's like a classic lupus presentation in a young female with a bad manifestation, which is potentially lupus nephritis. So she's admitted to, tertiary care, University of Miami, wonderful hospital. They get all the subserologies that reveal low complements and high NA speckled pattern. Nephrology corroborates the urinary sediment that is active, and she's treated for new onset lupus with likely nephritis. She's pulsed while you wait for the biopsy. The biopsy comes back and she has class 4 and class 3 with high disease activity and low chronicity score. And to be fair, this is kind of like the perfect scenario when you catch someone with a relatively, new onset disease, gets to a high clinical care center and gets a kidney biopsy, gets treatment right away. And you would think that this is a patient you can actually make an impact in their long-term outcomes of lupus. But to be fair, we are here, in best case scenario, following the 2024 guidelines of lupus nephritis, and we give what we give. This is our algorithm where we have new LN, we pulse, we can give hydroxychloroquine, which is our mainstay treatment for lupus. We work with nephrology to add, an ACE inhibitor when the creatinine is stable. And then we start over their immunosuppressive therapy, which is now like what we're calling triple therapy, which is like high dose steroids, mycophenolate or cyclophosphamide as a first-line agent, plus the addition of these new drugs that have been recently approved um, for lupus nephritis, which is belimumab or a calcineurin inhibitor, such as voclosporin, if there was a class 5. And this is all very much like, 2026 standard of care therapy and what we can offer this newly diagnosed patient where you could potentially think that we will do good. And to be honest, we're not really. Even now, in the latest and greatest treatments, we are basically failing a lot. Or like these are two very impressive studies that show that actually complete renal response, and this is what we measure in terms of proteinuria at 12 months and normal creatinine, where someone really has recovered from lupus nephritis to 100% where we can guarantee long-term outcomes is really poor. It's like 30 to 40%, even with the addition of um, anti-CD20 therapies such as belimumab or voclosporin therapies. And we know that roughly prior these studies that even we know that 50% of our patients are going to fail treatment with MMF, and we're going to have to switch them to cyclophosphamide. But we still don't know which are these patients. So we do what we call this pharmacological wheel of fortune, where we're like, okay, we go through all algorithm, but really it might take one or two or three trials to align the patient's biology to the mechanism of action of a drug to be able to finally really get into remission. And this is the case in most of our clinical scenarios, not just lupus nephritis, but this is like what we're dealing with till this day in clinical practice. And what we've basically came to realize is that the molecular heterogeneity of a phenotype. So here on the right, you can see an image of the hand, like clearly showing swelling of the MCPs and PIPs in particular. So inflammatory arthritis, inflamed synovium. And in the table, you can see that there's, as you say, so many ways of getting, like, so many roads to Rome, in the sense of like you can get to the same phenotype that looks exactly the same clinically. And it can be so different. It can involve like a variety of very distinct cells, from fibroblasts to plasmablasts to neutrophils to T helper cells that have a complete different cell of markers and complete different location in the synovium, and really complete different mechanism. So, you can understand why we've been very frustrated because we've been classifying our patients clinically all this time. And that's how we classify and diagnose lupus and RA and all our diseases. But we really are compounding so many different molecular pathways in a single phenotype, and that has really clouded at least our treatment and clinical trials, to be honest. So we're at the stage in rheumatology where the community has realized that we really have to redefine diseases by molecular taxonomy that will really try to help us align, a patient with the right treatment and the promise of, again, personalized medicine, and really stop doing this pharmacologic Russian roulette. And we've basically turned to other things that can help us. And this is where the wheel gets really bigger and bigger, because we have so many layers now of data that are coming into light with the promise, again, of trying to really define diseases from a molecular standpoint. So I'm just going to really start with genetics, which is the first one. And genetics, now we have 30 or 40 plus years of experiences with GWASs. And again, we thought when the human genome was sequenced that, this was going to be the end of diseases. And, we've all gone through a little bit of the hype and then the kind of not hype of, well, what happened with all these genetic studies and how can we use them. So I want to explain a little bit where we are in terms of genetics, particularly in autoimmune diseases. And the first thing is like I just wanted to really remind people of like what is a GWAS and like why it hasn't really been as, the holy grail as we thought. So to start, it's basically done as, they've mostly been done with arrays that have like placed little tags around the genome. So doesn't cover the whole genome, where you type a SNP that basically is like a marker for variants that could be all in the same chunk of DNA or chromosome, and that's something called linkage disequilibrium. But once you genotype this variant, you really don't know if that variant is actually causal. It could be any variant in that chunk of DNA that is tagged in that area. So when you find map, you actually can see a lot more signals, and you never know which one is actually the one causing disease. And this is an example from one of our, GWAS studies here in the lab, where you can see the top signal or the tag signal in the GWAS study is the top one. But when you look, there's all these potential variants underneath it that could way be like the actual causal variant. And it gets really nitty-gritty to really try to tease this apart. So for the most part of the signals, we really don't know if they're actually the disease causing SNP, and it really it's a mind-boggling work to try to really decipher each of these SNPs in the context of every GWAS that's out there. The other assumption that happened when the GWAS era kind of started is that, well, they said we have to pick some variants in these arrays to look at. So the assumption was, let's look at common variants. So what we assume that are going to be present at least in 10% of the population. So in having that assumption, they basically deleted a possibility of looking at rare variants. And rare variants are the ones that happen in like less than, 1% or 2% of the population, just because they thought it was really not worthwhile looking at these rare variants. But what we've known is that these rare variants are actually the ones that have like a really high effect size and are most likely to be pathogenic. Meanwhile, the common variants are the ones that we're detecting in GWAS. They each have like a really slight odds ratio, a ratio of 1.1, 1.2, really low effect size. And, someone can have like a flavor of all these like small disease variants that each of them have like a little, higher odd risk. And again, we don't really know exactly what to do with that, let alone know if they're causal or not, et cetera, et cetera. So, there's a lot of limitations with the GWAS. There's been 183 risk loci identified for lupus, 124 for RA. Again, we don't really know if the SNP is actually causal. And we really also just the array methodology just ignores a lot more that can be happening with DNA that, can actually be causal in terms of risk, which is deletions, insertions, variations in the copy number, et cetera. So we're seeing a lot of important genetic information. And, we've learned that these diseases are polygenic, with each variant having a minor effect. And it's really hard to really summarize what each of these things are doing. And also, we've been also learning that most of these variants, we're going to actually learn what they're doing when we see them in the right context. So, if you have a cell type with a variant, then you simulate the cell, and then it's when you actually see their effect at the level of the transcriptome, et cetera. And there's very few experiments that actually take a variant all the way through and try to put them in the right context to see actually how that variant actually operates. So it's hard. It's really hard in the area of functional genetics to be able to do that. And I think we're learning more that we have to really integrate these signals to really understand their effect, and not only at the level of multiomics, but also now at the tissue level. And I'll speak more about that. And then lastly, just because GWAS, again, you had to pick the variants in the arrays that you were studying. And the knowledge has always been in genetics predominantly from populations of European descent. The arrays were heavily biased to really detect variants in European populations that are really not the same because of this block. I was trying to explain a little bit of these chunks of blocks of DNA. They vary according to population. The linkage to disequilibrium is very different according to population. So it really is not applicable to one population to the next necessarily. But in any case, we are in the era of also there was a big hype about polygenic risk scores, and there is something to be said, and I don't want to necessarily glide through this, but a polygenic risk score is when you actually add all these GWAS signals in an individual, when you test for them, and then you summarize them, and then you weight them by the effect size of the GWAS studies, and you get a score. So it's a nice way of like conglomerating all these signals into a manageable number that can tell you something about the overall polygenic risk score of a person. And it has many caveats, but mainly the fact that it's been mostly driven by one population and not really assuming anything else. But having said that, after everything I said, it has been shown to be powerful. So now in, for example, type 1 diabetes, if you look at polygenic risk scores combined with HLA typing, you can actually predict the children are actually high risk for getting type 1 diabetes. And this is being used for screening and prevention trials. So that's really good. And in RA, also it has been shown that, if you have a high PRS, you're basically this at-risk population, and they're using that to do some trials of prevention in RA. And higher PRS have definitely been associated with being seropositive and worse disease, et cetera. So it is capturing something. In lupus, I found this paper that was recently published really interesting because it was a paper trying to link air pollution exposure and incident lupus in the UK Biobank. And what I want to highlight is not actually the effect of air pollution. This is the hazard ratios for exposure to PM 2.5, which is a measurement of air pollution. But if you have a high genetic risk calculated by a PRS here in the low side of the x-axis, your chances of getting incident lupus are pretty high. And this is, a very large population in the UK Biobank, which, it still shows that we are capturing at least people that have a high chance of developing disease. And this might be, at least we haven't been able to use the PRS score beyond that. But I think it would be great to start learning more about the use of PRS in not only diagnosis, but also outcome and, of course, treatment response. But it has been shown to be powerful. And again, this is just my little 2 cents that we really need to complete the genetic map for this to be equitable. This is a commentary about health disparities and how, polygenic risk scores are going to further enhance health disparities, because if it's just really driven the knowledge by one population, it really isn't going to, benefit everyone. This is just a very explicit diagram showing the lack of representation of other populations in most GWAS studies. And this is just in 2018. I think it might be changing, but not a ton. And because Miami and because of my background, I want to say like the Americas, like in terms of the population of the Americas, are extremely like there's almost no representation in most of genetic studies. And in rheumatology and lupus, particularly, we know that the population in South America, North America are like highly susceptible to bad outcomes and bad disease, and we really don't have that genetic architecture in these populations, let alone for the African American population, that we still don't have that data. So there is a lot to be done in terms of just making sure that we really have at least the genetic architecture covered for everyone, not just a couple of populations. And this is like an effort. I just want to mention that we're trying. This is a preliminary study really looking at the Peruvian Genome Project, which was a wonderful effort by the Peruvian NIH to go and do genetics in indigenous populations in the Andes and in the jungle, like truly indigenous. It took them many, many years, and they published their findings. And these are people that are healthy, so caveat. But we were just interested in the actual frequency of alleles have been associated with lupus risk. In this population compared to, for example, 1,000 genomes, which again is people in theory healthy, so no one without immunity here. And what we found was when we calculated PRS scores, again, with all the caveats I just said, the native Peruvians, the non-admixed Peruvians, had the highest lupus PRS score compared to other populations in 1,000 genomes, such as in Africa, European, Southeast Asian, and East Asian, which is very striking. And actually, it kind of diminished slightly when we included admixed Peruvians, showing that there is differences in just baseline genetic risk. I mean, as always, these diseases, they're not monogenic. You have to have this combination of genetic predisposition and a trigger. But if your genetic predisposition is kind of running high, you definitely need your chances of actually developing disease are higher. And it's just a call that we really just need to study the populations that are sick. In our case, the Latinos and the African Americans really do deserve their good genetic studies. So that's like my little argument for that. But and then lastly for genetics is that I think we're going to enter a complete different era now that we're past from the GWA era to actual sequencing, and these technologies are allowing us to really go base by base, and just completely, not use the arrays with all their caveats and really have the whole genome. And the costs are lowering every month, so it's more accessible for researchers to actually do whole genome sequences. And not only whole genomes, but to actually sequence a whole genome, and you actually have to cut the genome in a lot of short pieces. But now there's technology that allows you to really literally sequence the whole molecule of DNA in one string without cutting, which is called long read sequencing. And this will really, for the first time, allows us a resolution of all the potential structural variants, facing of caplets, a lot of like really rich information that might be a lot more meaningful than just the GWA association findings that we've gotten in the past 20 years. And it can also give us direct measurement of epigenetic modifications that we'll talk about. So I think we're just really have scratched the surface of what we know about genetic risk and how it can actually be used for subtyping and really again, trying to really utilize this in a personalized way in rheumatology. So we're behind, but I think in the next 10 years we'll learn a lot more. So I haven't given up on genetics, basically. That's my 2 cents there. And just as an example of what this technology can do, GWA, we used to study like thousands of people to get a effect size and have a power. But now with sequencing, you can study like a couple of families. And this paper that came out a couple of years ago that was very impactful because it really described a new gain of function variant in TLR7 and in 2 families with lupus, and really have defined a whole different pathway that can help. It's now being developed as a therapeutic intervention. But it just shows that, when you have interesting family, you can do sequencing, you can actually find a variant. And that's really going to change the game of how we understand genetics and the role of autoimmunity. And hopefully can utilize this to really understand disease heterogeneity and prognosis. So I think it's going to be a fun era for genetics and in rheumatology in general. This is just a timeline that the more of these technologies are being used, the more quote unquote monogenic lupus are being described. And this is just going to be really interesting. I think all works together, but it just shows you an example of how much our understanding of the genetic risk of a disease is improving now that we're being able to do these assays. And speaking of which, there's been a big effort of doing whole genome sequencing in biobanks, as you may be familiar with the UK Biobank. In the UK, we have over like 500,000 participants. with, whole genome sequencing and phenotyping data and EHR data. All of us is basically trying to do the same and really enroll a multi-ethnic cohort with the same level of data. I think here we're still actually our lab sits like next to the All of Us lab, which is fun. But we're working a lot in trying to get just have the EHR data at a point where we can really trust the phenotype, so we can call lupus lupus. But I think we're excited about these big efforts, so we're going to really kind of try to answer a lot of these questions with real life data. So there's an exciting avenue there. So the next kind of layer I wanted to talk about is epigenetics, which is close and dear to my heart. Again, when I heard this as a resident, I was fascinating of the actual concept that we can inherit not only genes, but how our genes express themselves. It's a very powerful concept. And to a fair extent, it's very hard to study, but the concept is still fascinating. So what is epigenetics? And epigenetics really is basically all the modifications around DNA that makes it accessible or inaccessible to gene expression. And this basically is the diagram showing, here heterochromatin, like super tightly packed around nucleosomes and, chemical additions, impossible to read, versus this like really nice loop of euchromatin that's like really relaxed where all gene expression is happening. And everything that is involved in like this translational change between euchromatin and heterochromatin is called epigenetics. So it's a lot. And DNA methylation is one of these modifications, and it's easily studied. And this is the one that I'm going to focus on, but I just want to say that there's a lot more to epigenetics than DNA methylation. And the concept here is that these epigenetic modifications are highly dynamic, as you can imagine, and it's basically they're maintained by a series of enzymes that not only have to like keep the epigenetic signature of a cell intact, but it has to do it every time a cell divides as well. And just keep it there in terms of the lifespan of the cell. So all of these cells enzymes are very susceptible to the internal environment of the cell, such as pH or metabolism or radical oxygen species or vitamins or nutritional status. So the idea here is that what if these, mechanisms fail and then there's a drift, like an epigenetic drift where for some reason the quote unquote epigenetic signature was not maintained, but there was an invariant like signature there. And the interesting concept here is that once this aberration happens, you can then transmit the same gene expression pattern in further cellular division. So it's something that you can inherit or can be transmitted over time way after the exposure happened. So it's a very interesting concept. It has been proven a lot in, for example, worms in the lab, where they can like do 20 generations, and after big exposure with heat or some in vitro exposure, they can see the epigenetic changes going over several generations. It hasn't fully been proven in humans, but it's very interesting concept of how to explain our diseases or how we use this to really also dissect diseases. So we've been measuring DNA methylation in our patients in our cohort in California and lupus, and this is just one example of what we're trying to do with these different areas of multiomics. What we did here initially was just to cluster our patients clinically with machine learning to see what type of flavors of lupus we had in our cohort, and we loosely defined three kind of classes of like mild lupus and two that were a little bit slighter severe than the other. Like, the severe two was like the pan-positive serologies, bad lupus versus the mild one, which had only skin and rashes. And we see that in clinic a lot, the whole spectrum. And then we looked at their methylation marks, DNA methylation, and we performed what we call an EWA instead of a GWA, and tried to see if there was any differences in methylation marks across the genome and these different subtypes. And we did. We saw a signature of 250 methylation marks. We call them CPGs. And this is a heat map showing clear view, I mean, the clearly obvious difference is between the mild cluster and the two other severe clusters. But there are some other differences where you can see red means in the heat map that it's more methylated and blue is like less methylated. So there's some differences in methylations in all these genes in the genome. And when we looked at which genes, they were mostly immune-related genes, which made sense, mostly lying in the interferon signature. So it was really intriguing for us to see these methylation differences in our cohort according to clinical subtypes. And we actually kind of followed this over time. We remeasured methylation 2 years after to see if this signature is still, hold, because again, the methylome is highly, variable. And we actually kind of honed in a set of CPGs that were very stable over time, that didn't fluctuate according to disease activity or medications. So it's very intriguing. So we have these potential, epigenetic biomarkers of disease subtype. And here we're trying to see if we can basically study these CPGs prospectively, and see if someone gets a recent diagnosis of lupus, can we get their epigenome and see prospectively what happens and see if we can actually prove this set of CPGs to actually be useful in clinic. So that's where we are with this idea. Um, the next layer I wanted to talk about, which probably you guys are more familiar with, is transcriptomics, which is gene expression. And this is just sequencing of RNA. And you can do this in pretty much every tissue, whole blood, in cells, and any tissue that you need, or you can get from patients. And it's more widely available. My 2 cents on RNA is that it's very, what you sequence is what you get in terms of that point in time. So you always have to take account into the concept, the context. Because it reflects like gene expression in the last 4 hours. But it has been a very powerful tool, not only in rheumatology, but in pretty much every field in medicine. Again, this is just an example of one of our papers looking at the transcriptome in different cell types in the California cohort. And what you can see also is different clusters of genes being expressed in different cell types according to disease manifestations. So you can definitely have associations of, different particular gene expression models with, some of the classification criteria. Again, these are all association testing, which are very intriguing. I think we're still trying to figure out, okay, how can we take these signatures to something that makes clinical meaningful sense, or at least therapeutic sense. And I think we're basically still kind of in the describing mode and trying to understand these programs in different settings and different cells, um, and taking it from there with a grain of salt. The technology now has shifted to single cell RNA-seq, which I'm sure also you guys have heard of. This is again collaborative work from the cohort of California, where I think is probably the largest single cell atlas in lupus. And what was interesting with this effort was that we really could actually define again the role of some genetic variants at the level of gene expression in particular cell types, where you can really see the effect of genetic risk at the level of gene expression. And this has been mostly like a resource because it's such an amount of data. And again, the study design matters. So this is just, a prevalent cohort of lupus patients. Again, this is a question like, when do you sequence? And for the most part, these were very stable patients and 40 years of disease with immunosuppressant therapy on board. But it's just giving us a flavor of what we can potentially get in terms of data. And then on the right panel, I wanted to show that now we're also shifting to basically in situ or tissue single cell expression. This is a picture of synovial biopsy where you can get histology and also get a single cell readout of gene expression of the cells in a tissue plane. And that will also be really super helpful. I think there's a lot of efforts doing this in kidney biopsies of lupus nephritis to really try to understand again this molecular heterogeneity and see if we can better target therapeutic therapies. So these are all new technologies that I think it's going to take some time to figure out how they land in clinically meaningful ways, but it's exciting. Um, I also, in terms of the transcriptome, want to mention this paper that it's now, I mean, not super old, but not from yesterday, but just shows the power of what we can learn with these tools. This is the PRIME study published in the New England Journal, and it's it's an N of 4 patients, so very interesting. But what they did is that they actually send them a kit to have at home where they could like basically do pinprick blood, which also requires sophisticated technology to be able to perform gene expression testing from a kind of pinprick drop of blood. But so anyway, so they had these patients do this every 2 weeks. And then they analyzed the data in 2 years' time, so they could have samples before someone with RA flared, before having an RA flare and after. And just using this gene expression data collected over several years, every 2 weeks, they actually detected a new cell that basically appeared in blood 2 weeks prior the flare that they coined the prime cell with a mesenchymal cell. Um, so just helping us understand actually the drivers of disease activity and like the genes involved that could be maybe highly interesting, or you could see that this might actually be clinically helpful. Um, one day when patients can actually monitor this and, oh, there's a signature, I'm about to flare, you could potentially do a few preventive things. So it's we're learning about how to use these technologies that are really interesting. Um, so let's see. What do I also have? Oh, the exposome. So the exposome, I mean, genetics is never isolated. We always know the context is very important, particularly in our diseases that are polygenic and, they're occur in adult lives. So the concept here is like you do have some genetic risk, but something else happens that triggers, to go to the other side of breaking tolerance. And the exposome is this really interesting term developed in 2005 by epidemiologist Christopher Wild out of frustration. Like, we need objective measurements such as the genome. Like, we need an exposome. We need to be able to actually understand all these environmental signatures of risk. And it's basically been coined, and I think there's a lot of efforts to trying to do that. And again, our lab tried to have a bit of an experience here where in a just technology is allowing us to do these things where it's interesting. We're using mass spectrometry. You can actually measure every single molecule that's in your blood, and you can really detect exogenous molecules. So in this case, pesticides, classifiers, flame retardants, all the wonderful things that we're exposed to daily. You can actually measure them with a high resolution mass spectrometry. So we decided to do this in the California study. Um, it was a very challenging. I think the technology is still not there. It's like 10 years of work trying to figure this out. But it was interesting. We could agnostically detect all these things. You get a mass peak, then you match them to a database and you get a name. And then you can basically see what you get in an agnostic way. And what we found was a signature of like higher pesticides in our patients than healthy controls. And when we looked in detail, we also found a lot of phthalates that were highly present in our lupus patients versus controls. Again, study design is key here. It was like, prevalent cohort. This is not causal. There's so many context-dependent things. When we found this phthalate signature, well, I had to learn a lot about phthalates, which I had no idea what they were. But so just to see the actual confounding here, phthalates are plastics that are added to all the enteric-coated meds. So basically, if you have any medication that's enterically coated, they put phthalates in it. Not that they're good. We know that they're bad. We shouldn't be doing it. Every time you go to the ER and you get an IV, like the whole lining is like covered in phthalates. So like you basically get exposed. So we're probably giving our lupus patients a lot of phthalates just to intersecting with the healthcare system, which shouldn't necessarily a good idea. But again, it was like interesting that we could capture these signals. And again, in the right study design, I think having the tools to be able to measure exposures in this like real throughput way could be really interesting. It could be maybe illustrative of maybe some things that could be really predisposing to risk. So we're investigating using this technology in samples in cohorts that have, for example, plasma before they actually developed illness, et cetera, et cetera. So we're looking into that. Lastly, this is not work from our lab, but I've been very interested in following the whole story of immunometabolism, the microbiome, and autoimmunity. And these are just three really interesting papers that I'm just going to briefly summarize. But we know that, for example, we're learning a lot more that the function of an immune cell is highly tight with the metabolic program of the cell. So if the cell is in like a glycolysis, like an immune cell is choosing glycolysis versus fatty acid oxidation, there's a chance that these programs are highly linked to the function of the immune cell. So you can actually shift the metabolism from glycolysis versus non-glycolytic pathways and actually change a Treg to a T effector cell. So it's a very powerful concept. Like also because we can potentially intervene in a significant manner. There's a lot of opportunities for intervention here, but we've been learning a lot about this concept of immunometabolism. And these are tightly linked because a lot of the metabolites that we get are from our diet, but not only our diet, but the diet that's also in this tight dance with our microbiome that decides to, digest food in a certain way according to the bacteria that you have. So it's all this very nice story linking diet, microbiome, and immune function that could affect autoimmunity. And I think it's like a complete different area of research that is very interesting. And again, here the concept is that you have this priming, metabolic priming, that then gets exposed to maybe dysbiosis or a breach in intestinal translocation, causing potential cross-reactivity with antigens and the whole downstream effect of autoimmunity. But I think this is just to mention that we're just like getting a lot more information of different omics, and it's been really interesting to see. So there's so much data. And I think as a researcher trying to do this, the challenge here is like, how are we going to make sense of all the data that's been really produced in all these different labs? And I think there's not a single way to do this. Every lab does it in a different way. There's no consensus, but we are now really getting a lot of boost from AI, which I'm not in disagreement. I think we probably need this, but there's a lot of black box, like we don't know what these things are doing. How are we going to reproduce our findings? So I think this is going to be the main challenge of really trying to understand how all these things work to deliver in a clinically meaningful way in clinic. I wanted to leave you with a few examples where this is working. So we have some evidence where when you use a molecular classification, we can actually learn. And this is the evidence from the R4RA trial published a few years ago when they basically looked at RA patients that had failed TNF therapy, and they looked at their synovial tissue and also gene expression in blood and try to understand if any of these molecular profilings could distinguish people that were going to respond to either tocilizumab, which is an IL-6 medication, versus rituximab, that is an anti-CD20, complete different mechanism. And what they found was really interesting is that actually the gene expression signature was more powerful in predicting response to tocilizumab versus rituximab. So that's really great. I mean, if we can potentially figure out how to bring this to clinic at one point, but this is basically the dream, right? Of getting synovial tissue with relevant information that will guide at least inform therapy decisions that are out there already. This is another trial, the STRAP trial, that looked again at synovial biopsies and tried to identify signatures of response. And they described again two really powerful signatures with response to etanercept and also tocilizumab, two of our main drugs for RA. Proving that if you get actually some, molecular information, you can have some form of guidance as to what therapy to pick. So I think these are really powerful examples. In lupus, I do think that we have at least this. We have one medication called anifrolumab. It targets an anti-interferon receptor alpha beta. It got recently approved for lupus, and you know, long story short, if you have a high interferon signature, not all patients with lupus have one, but if you have a high interferon signature, you're most likely to have this response rate of anifrolumab, which is the Kaplan-Meier result here. The differences between placebo and anifrolumab are clear, but if you don't have a high interferon signature and they give you this anti-interferon medication, you really don't have a response that's different to placebo. Shocking. But I think it just highlights, like, if we have this information in clinical setting where we could actually make these decisions, it would be really wonderful. We don't have them just yet. And lastly, I think I've talked a lot about omics, but I think honestly, I feel like we're also missing several layers that try to understand how a patient is gonna, go well. We know that nothing of these matters if the patient is poor and does have poor access to care. So, I just want to really acknowledge that we really do have to determine, like, in all these models, incorporate not only the social determinants of health, but like also like what that person can and cannot do in terms of health habits and the whole cosmology of illness, like how they're interpreting their disease and all these other layers that really do influence outcomes and treatment response that we're, I think, missing in most of our biological framework of research. So it's just very complex. Every single patient is their own universe. So that's why it's really beautiful in clinic, but very challenging in terms of research. But I think there are some headways, and this is kind of like the ideal scenario of what I see the future. I think RA is probably going to be the first disease that gets there, where you have a new diagnosis of RA, you get a synovial biopsy, you identify all the cell types, all the gene expression programs, you actually do some in vitro work with the cells, you really understand the mechanism, and then you prescribe the right therapy. And I think that's probably going to happen in RA, I think, sooner than any of the other rheumatologic diseases that I can think of. And with that, I just want to thank all my collaborators and happy to take any questions. Thank you so much.
Thank you, Dr. Lanata. That was a really instructive grand rounds. We appreciate the clear slides and the discussion. It seems as though rheumatology has gone from an understanding the taxonomy of the antibodies and all the antibodies that we use to help us diagnose, and now we're having another layer of the molecular components, the molecular signatures that help us understand, maybe with a little bit more insight, to potential therapeutics. So my question is, and you sort of touched on this in the last couple slides in terms of RA, how close are we to a real world use of the combination of all these things? And you say RA will be the first one, but really, how close are we in terms of realistically being able to apply this in our clinics?
Oh my goodness. I have no informed informed data to answer. I want to give RA like 10 years. I want to say that in 10 years we'll get there. And I think yeah, lupus. Oh, I think in 10 years we'll have a better map of where to go. But I think, we'll be better off in 10 years than we were now. We joke with our oncology colleagues because right now is like we would be treating every cancer with the same chemotherapy, with the same regimen. That's how we're doing rheumatology these days. You have lung cancer, we're gonna give every lung cancer the same therapy. And it's literally that's how we're doing rheumatology. And I think we'll hopefully follow behind oncology in their level of precision therapy. But I'm gonna give us 10 years to be able to sort
Okay, well, we're going to invite you back in 10 years and we're going to
of.
see.
That'll be great.
We won't hold you to it. That's fine. Dr. Campos, you have a question in the chat? You can unmute yourself.
Oh, yes, thank you. As I wrote there is a theory that patients may have prior trauma and gets embedded in the subconscious and dysregulates the PNI axis. And that's a predisposing factor to develop many autoimmune diseases. I wonder if you can comment about that.
Yeah, no, thank you. I didn't have time, but no, for sure, that's a big elephant in the room. I feel every time we talked to particular patients with lupus, we did look at trauma in our California cohort. We looked at adverse childhood events, which are very traumatic experiences as a child. We looked at just any traumatic event, and the prevalence were through the roof. I think 60% of our cohort had an adverse childhood event. Is just ridiculous. It's super high. And I think, as you said, the translational evidence is really there, linking stress with immune dysregulation and autoimmunity. And I have several colleagues at UCSF that are looking at that in detail. Clinically, I think it's a challenge. I think I personally don't feel like I have the tools in clinic to open that Pandora box. But I think the day that we would, I think it will improve outcomes. And as I said, I think there's many layers that we're not really integrating, and how the disease process can go better if we take care of all these other layers that are clearly contributing to disease activation and outcomes. So thank you for that comment.
Dr. Lakakis. I'm sorry, Dr. Lakakis.
Yes, sir. So if high lactate induces Tregs, in lupus, I believe the lactate is high. Why the Tregs don't decrease the inflammation?
I'm sorry, I didn't catch the end.
Why the regulatory T cells that are
Oh.
by high lactate not suppress the inflammation?
Oh my goodness. Again, I think lupus is such a hard one because even though you tackle maybe one pathogenic mechanism and the highlighted story, I think there's also a lot. I didn't have time to go over the new findings of all the mitochondrial dysfunction, and I think there's a lot of interest there. I think maybe you can tackle that, and maybe in a subset of people that might be the right thing to do. But you also have to account for the rest of other pathogenic pathways that are dysregulated. So I can't answer as to why it doesn't work. I think it might maybe work in an interesting subset of people. I know, like IL-2 therapy and just Tregs. I mean, there were some studies at UCSF where they actually infused Tregs in lupus patients and N of 2 or 1, and they had some good results just trying to get the Tregs to do their job. But again, it's not going to work in everyone, and that's the challenge here.
Dr. Contreras.
Yes, very nice talk. Now, with all this molecular printing in lupus, do you see the same type of printing, let's say in the synovial, in the kidney, and the same patient, or that's different? Because lupus affects so many systems, and I wonder if the molecular printing is different in each system.
Right. So the AMP Consortia, which is the Accelerated Medicine Partnership here, led by NIH, really is tasked to do that deep phenotyping in different tissues for lupus and RA, and they're still working on, but their mission is can we really get to tissue phenotyping? So they're collecting kidney biopsies. We know blood is an important compartment, but it's not where the action might be in lupus. Maybe all the immune cells are actually infiltrating tissues and causing damages there. And to your point, we might be looking at the wrong specimen. So I think there's a lot of effort in getting tissue. It's hard. That's why I feel like RA is going to be um, uh, it's easier to get a synovial biopsy for research, and clinically it's just like easier to get tissue from RA than from lupus, specifically because there's so many, you know, organs that we can't really go and take a tissue sample. But it's a very important question. I think a lot of consortia are trying to solve that, like where does this phenotyping and profiling matter the most? What is going to give us the better bang for a buck? So I think we're in that process of trying to understand that.
Thank you very much, Dr. Lanata, for an excellent grand rounds. I want to remind everybody that next week we'll be rejoining each other in person and hybrid option as well over in Rosenstiel. So I look forward to seeing everybody. Lunch provided as usual. I'm going to remind everybody to check the chat for the link for CME and MOC. And thanks again, Dr. Lanata and Dr. Chung, for inviting our excellent speaker today. And everybody have a great day and take care of yourselves and each other. Thank you so much.